Okay, so in Chapter 9, we are going to be talking about cellular respiration. This is how animal cells go about getting their energy. A lot of bacteria actually use these same pathways for cellular respiration, and most non-plant eukaryotic cells will use these pathways for cellular respiration. There are some differences with some algae, and there are some differences in some of the bacteria, but this is by far a very common pathway that you're going to run into. So the catabolic pathways are going to end up yielding energy because they're going to oxidate organic fuels. So catabolism, we know, is breaking things down, and it's the potential energy that's going to get released in these exergonic reactions. So exergonic means you're going to release energy. And there's three different ways that you can do, or it's actually two main pathways in here. You've got fermentation, where you're going to partially degrade the sugars, and this occurs without the use of oxygen. And then you've got aerobic respiration, where oxygen is going to be used. And aerobic respiration and cellular respiration are the same thing. They are also known as aerobic metabolism. It got called respiration because of the association with breathing in there when they were looking at it in animals. But aerobic metabolism is actually probably a more accurate term, since it does occur in cells that don't actually, it will occur in single-celled organisms that don't have lungs to do what we think of as breathing in there. So you start out with your organic compound, and then you add oxygen. So it will be in the presence of oxygen. And then you get carbon dioxide, water, and energy released. So the specific example we use is glucose, and most things are going to actually join the same pathway that glucose is on. So it's a good one to study. Glucose is your C6H12O6. So for one glucose, you're going to need 6 units of oxygen, and what it's going to yield is 6 carbon dioxide, 6 water, and then your energy in the form of ATP and heat. And the delta G of that is negative 686. So because it's got a negative delta G, it is a spontaneous reaction that would naturally occur. You're not going to have to put any energy into it. So the catabolic pathways don't directly perform cellular work. You have to convert that energy into ATP, and you use the ATP to fuel the work. So ATP acts like a chemical drive shaft, and therefore you regenerate the ATP. So the ATP is like going to work and getting your paycheck, and then you use the paycheck to pay for other things in there. So most of this occurs through what we call redox reactions, or they're sometimes called oxidation and reduction, reduction oxidation, whole combination of that. Redox is the way I learned it in chemistry. It's the shortest name. I like that one the best since it's the shortest. And what all of these involve is transferring electrons. So you're going to move electrons from one substance to the other. If you're going to move electrons and remove them from something, something else has to gain the electrons. So these things have to occur together. You can't reduce something without something else being oxidized as a source of the electrons. So the oxidation part is the loss of electrons from one substance to another, and the reducing part is the addition of electrons from one substance to another. The reducing agent is the electron donor. The oxidizing agent is the electron acceptor. So when something is reduced, it has gained electrons. And the way that there is this thing, this mnemonic oil rig, oil is oxidized is lost, reduced is gained. The way I remember it is with reduction, it's a gain of electrons because you're gaining more negative charge, so you're making it reduced whole value in there. It's kind of a hokey way to have to remember things in there, but it is what it is. So this is an example here showing where you've got this reaction going, and part of it is being oxidized, part of it is being reduced. So this is showing here where you've got part of it with your sugar becoming oxidized, and then things being reduced here in the reaction of cellular respiration. So this also shows it here with methane becoming oxidized and the oxygen becoming reduced down to water. So as things lose electrons, they're going to lose potential energy along the way, and the energy state is going to go down, which is what gives it the negative delta G and makes it spontaneous. So when we do this in metabolism, instead of completely oxidizing glucose in one step, you're going to start out with a series of 10 steps to do the process of oxidation in there. By having it be in 10 steps, it's going to allow you to have a lot more control. You use a lot of the intermediates that are made along the way. But to do that, we're going to have other things that are going to have to hold electrons and move them along the way. And one of them is the NAD+ in the electron transport chain. So NAD is nicotinamide adenine dinucleotide. Everybody just calls it NAD. And then the reduced form of it is NADH. So you're going to alternate between those two states of it. Basically it's a bank to be able to move electrons to and then take them off of. So it's something that's used in there. And you're going to see that a lot in that reaction. There's the enzyme that's going to work with it, the dehydrogenase in there. But NAD+ and NADH are the most versatile electron acceptor that you see in respiration, so you're going to see that used quite a bit in these reactions. So when we end up reducing the oxygen and to make the water in there by reducing the hydrogen, that's actually a very explosive release reaction. It's got a lot of energy in it. So in turn for letting those things come together, we're going to take that energy and use it to glue phosphates on to make ATP. So that's why this reaction works so well, is this releases a lot of energy, and we use that energy to make our ATP that we fuel other things with. So this is just showing the NAD, what it looks like. If you need to see that, and then it gets converted to NADH. For this class, you're not going to have to recognize the structure of it. You won't even need to really know the name of it, the full name of it. NAD and NADH are good enough in here. And I highly doubt you'll have to use the full name of it unless you go on to do a lot of biochemistry stuff. So this picture here has a nice overview. You might want to mark this picture as something to come back to when you're looking at this, because when you go to study this, by the end of today, your head will be spinning and you'll be like, what have you loaded me with for the weekend? When you go to study this, I'm going to strongly recommend you study it in small little chunks. When we break down glycolysis, study that chunk, then study this piece, and this piece by themselves and let them sink in. This is not something where you go, well, I'm going to save this till we get closer to the test. This is one of those topics that you're going to want to spend at least 20 minutes a day looking at one of these things, writing it out, playing with it however you need to get it to sink into your head. Because it's not one of those things that anybody can explain it to you, that it will just go in the first time. You have to work it out in your head. It's like you put it in and you got to shake it until all the rocks fall down into place. And this one just happens to take a lot of shaking to do. So glycolysis is going to be the first part of this. It's going to occur in the cytosol of the cell. During this process, we're going to end up splitting glucose in half. And when we split it in half, what we end up with is 2 pyruvates. That's just the start of the cycle. When we do that, you're going to get a little bit of ATP. And when you're breaking it in half, you're going to be moving some electrons from doing redox reactions. So they're going to get carried on this NADH. So it's kind of like saying, all right, I broke this, I got some energy from it. Some of it goes into this pile, some of it's going to go over into this pile. This is just going to be a delayed form of energy. So the citric acid cycle, or Krebs cycle, it's also called the TCA cycle. In the textbook for this class, they like to call it the citric acid cycle. I am not sure what they refer to it as in the 241, 242 textbook, which term they use, but just know these are all the same. Krebs cycle is the other really common name. I will use them interchangeably because all the other textbooks I use call it this. So this step here where you take pyruvate and you convert it into acetyl-CoA, what you're doing with this step is you're just getting it ready to go into the citric acid cycle. This is going to occur in your mitochondria of your eukaryotic cells. If it's a prokaryotic cell, it's going to occur in a membrane fold in the cytosol. Here you're going to completely oxidize the glucose to carbon dioxide. So in doing that, you're going to get a little bit more ATP, and you're going to have to carry some more electrons on, NADH, and then we have another electron carrier, FADH2. And then finally, in the last step here, the electron transport chain, we are going to deal with all of those electrons that we've made here, and we're going to let these electrons go to a happier place, and in exchange for doing that, they're going to end up giving us the energy to make more ATP. So that is your big overview picture of what is going on in here. So if you get confused and you're like, why am I doing all of these parts of the cycle? This is your big picture that you want to come back to. And if this picture can make sense to you, it actually, if just understanding this part of it is probably about 50% of a biochemistry class right there. So in that last step in the electron transport chain here, where we move these electrons and we do this oxidative phosphorylation, we talked about when you combine these hydrogens and the oxygen to make water, that this is an explosive release. In doing that, we take that, and it's showing it here with little turbines. If you've ever gone and taken a tour of a dam on a river, what it basically has you do is you go through and it controls the water and it spins these turbines and it spins inside of this thing and it's magnetic and it generates the electricity. It is taking the energy and using it to make the ATP. So, and it's going to just do it in multiple steps. Since this is an explosive reaction, you get to make a lot of ATP from this in putting these together. And when you move more electrons, you get a bigger hill to go down, more ATP is made. And we're going to break all of these down into little details, but I'm trying to give you the big picture before we go into that. So for each glucose that you're going to completely oxidize to carbon dioxide in water, you end up with a net gain of about 36 ATP. So you have 38 total come out. You end up having to invest 2 ATP to get the process started. So that's where the net gain of 36 comes from. So this picture here is another good one to refer back to that you'll probably want to highlight or at least mark in your notes to come back and look at. So when we look at glycolysis, which is this first phase that we're going to do, you have your initial phase where you're going to have to put some energy into it. So if you go to start a fire, starting maybe your barbecue, you don't just walk up to your briquette barbecue and say, burn. If you do and something burns, you probably shouldn't be here. You could be making lots of money other ways in this world. You have to put a little bit of energy in. You light a match to try and get it started in there. And you have to actually really light that match and put in a couple to get it started. It doesn't start the first time, so put some energy in. Your barbecue gets going in there, and you're going to start to get some energy back out of it. So you're going to get the ATP back. You put in 2, you get 4 out, you're going to have a net gain. Also in the process, you're going to lose some heat. So you can think of that heat being like these electrons. But instead of it being lost to the world, we capture the electrons on this NADH, and we're going to use those later on. What you end up with is 2 pyruvates and 2 waters out of the deal. So down here, this is your summary of what all these reactions are going to give you. Glucose gives you 2 pyruvate and 2 water. You have a net gain of 2 ATP, and you have 2 NADHs that are formed in the process. And I would highlight this part of the box right now as something you're going to want to know. So in your notes here, this written part is an explanation of what is going on in the picture. So you can always refer back to it. You're not going to need to know all of these individual steps, but we're going through the individual steps so that when you look at that summary, it makes sense to you as to why that happens. And that's how I would encourage you to look at it when you're studying it at home. If you can look at this and follow it and go, okay, now that makes sense to me, how this happens, it will stick in your mind a lot better than just saying, oh, memorize this. So what we start out with, this is your steps that are involved with investing energy, and you start out with your glucose here. And it's just that 6-carbon sugar. It's showing it in a ring. And this is like your barbecue briquettes. We need to get your barbecue lit. So you throw in the first match. You're going to take the 3rd phosphate off of this, so it goes from being ATP, which is triphosphate, to diphosphate. And you put it on to carbon number 6, and that's why we call it glucose 6-phosphate. The 6 means it's on carbon number 6. This is just the enzyme that is going to control the reaction here, hexokinase. So here we have glucose 6-phosphate. It's kind of smoldering a little bit, but it's not really burning yet. And it rearranges itself a little bit to be fructose 6-phosphate. However, your barbecue is still not going. You're not going to cook any meat on just a little tiny smoldering fire. So you light another match to put in there. And we're going to take the 3rd phosphate off of there. And it's going to go over here. So now you have fructose 1,6-bisphosphate. Meaning you've got, bis means 2, so sometimes you'll see this is diphosphate. You've got a phosphate on carbon number 1 and a carbon number 6 of fructose is what that name means. So now your fire is going, and if you've ever watched wood burn in a fire, sometimes you'll notice the log will just kind of split in half in there as it's burning up. This is going to have enough energy in it that it's going to split in half. So one of the things it's going to make when it does that is dihydroxyacetone phosphate. The other one is glyceraldehyde 3-phosphate. So these two are interchangeable, but because of equilibrium, if you take away this one, and it's trying to keep half and half of everything, this one's going to keep getting converted into this. This is the one that moves on to the next step. So all of this is going to get converted into this. This step here is possible, you just never see it happen because you always continue to use this one. So far makes sense. You got to put energy into this to split it in half. Okay. So now we come to the part where we're going to get a little bit of energy back out of this. So that glyceraldehyde 3-phosphate is going to, your glyceraldehyde 3-phosphate from right here is going to come in at this step here. And we add some inorganic phosphate here This just means this is phosphate from other places in the cell. Basically, it's not coming from ATP, it's coming from other places. So now we're going to have 2 phosphates on here again, 1,3-bisphosphoglycerate. And now it's got enough energy in there, it's going to kick out some heat. And what we're going to do is take one of these phosphates off, put it on a DP, and you're going to get ATP out of it. The reason you're seeing 2 here is when you split that glucose in half originally to get the glyceraldehyde 3-phosphate, you end up with 2 halves. Both of them end up eventually as glyceraldehyde 3-phosphate. So you have 2 of each of these things along the way, and that's why you're getting 2 ATP here, is you're getting 1 from each half. So that's where the 2 comes from. We're going to rearrange that a little bit more. So it's going to go on to 3-phosphoglycerate, rearranging it again to carbon number 2. So it's 2-phosphoglycerate, rearranging it a little bit more to phosphoenol pyruvate. And we're finally ready to get off some energy again. We're going to pull off another phosphate, put it on ATP. Because you have 2 of them, you get 2 ATP. And we end up with pyruvate at the end. So making sense so far? Can follow this on paper? Not necessarily reproduce it, but can follow it on paper. So that is the first step, glycolysis. So all of that is what this box is when you come back to this picture. I know, this is one of those things that if this is the first time you've learned about this, you'll walk out of here going, what have I signed myself up for? Good. And eventually it will seem like not a big deal to go through it anymore. You will have this covered in every biology class. It's just, eventually you can't help but get it. So here's this pyruvate that we made from glycolysis. And remember, we're going to end up with 2 of them here. And we need to put this thing inside of the mitochondria to go on to the next step. So it's going to go on this little transport protein. And we're going to pull off a carbon in there. And with it, we're going to take 2 oxygens to make carbon dioxide. So what do you think happens to this carbon dioxide? We're thinking humans now. We're not too worried about what the single-celled stuff does. Oh, what do you think happens to it? What do you do with carbon dioxide? You exhale it. So that carbon dioxide that you exhale, this is one of the places that it comes from. So you've pulled off this. We went from having 3 carbons in it now down to 2 carbons. And when we pulled that off, we got more NADH, so we've moved more electrons. And this coenzyme A gets added in. And so now you've got your 2 carbons here for this acetyl-CoA. And this is going to be what goes into the next step. So you'll notice that these pictures are kind of even color-coded to go with this one here. So the kind of greenish one, now we're at the kind of light orangish one, and now we're going into the darker orangish one. So again, this is written out details that you can refer back to. I personally think it's just easier to look at the picture and only go with these if you're really stuck with something. So we've got this acetyl-CoA, and you actually have 2 of those from 1 glucose. This has 2 carbons on it, and we've got this oxaloacetate that was left over from the last cycle. It's got 4 carbons on it. So you go 1, 2, 3, 4. You put this on there, 4 plus 2. Now you've got citrate that has 6 carbons on it. And we're going to rearrange this citrate a little bit to make isocitrate. So remember, iso just means an alternate arrangement of something. So everything in here is still the same as citrate, it's just in a slightly different order. Now we're going to pull off another one of these carbons, and we're going to combine it with oxygen. So the reason this is considered oxidative metabolism is if you don't have oxygen to attach that carbon to, you're not going to pull it off and it isn't going to happen. So you pull off a carbon. When you do that, you're going to release some energy and some electrons. So you're going to make another NADH here. And now we're down to 5 carbons, which is alpha ketoglutarate. And we're going to pull off another carbon and get carbon dioxide. You got to stick it with the oxygen. So at this point now, we have broken off both of these carbons that we've added into this cycle. We're down to succinyl-CoA that has 4 carbons in it. More NADH is released. So you've got your charcoal briquettes in your fire in here, and they're starting to crumble and break down so that they're going to end up that pile of ashes in the bottom. So as we're going through all of these steps here, we're just continuing to rearrange and crumble and break down and release that last little bit of heat. So your succinyl-CoA, it's going to get rearranged, and you're going to have a phosphate get pulled off. It's going to make GTP, which is very similar to ATP, but it's right away going to get converted into ATP here. So it just doesn't fit on the whole screen. This ADP will end up making ATP. And you end up with succinate, still 4 carbons. We're going to rearrange this a little bit more again, release some energy and some electrons. We're going to stick the electrons on FADH2 this time. So this is like a currency to move electrons and energy. So you had ATP, which is like your dollars. Here you got some Canadian dollars, now you've got some euros. It's just another currency, another way of moving things in there. So your succinate becomes fumarate. Fumarate's going to get rearranged to make malate. Malate's going to get rearranged to make oxaloacetate. And release a little bit more. Here is NADH, some more electrons get moved. In the process, we are back to this oxaloacetate that we can put more acetyl-CoA on. So if you think about it, when you exercise and you go for, say you want to go for a nice easy run or a jog, there's kind of a maximum speed that you can go where after a while you just can't breathe any harder in there. The maximum amount of oxygen you can take in. That maximum oxygen you can take in is going to limit how fast you can do this cycle. Therefore, determining how much energy you can produce, which is going to end up determining how fast you can run. So the more oxygen you could take in, the faster you could do this cycle. However, you got 2 holes in your nose, 1 in your mouth, and that's all the more that's going to come in. Also, you only have so much blood to move oxygen into there. So that's why this is limited by your oxygen intake. So imagine you go up to Mount Rainier and you're up at Paradise up there, and you decide you want to run up there. You're going to find it's not so pretty when you go to run, that you're going to get tired a lot faster because you're going to get a lot less oxygen from the air, and you can't do this as fast as you want to. So that's at around 5,000 feet. So we'll say we drop you off up at around 10,000 feet. So if you've watched any movies with climbers, you'll notice they show them walking really slow like this. And it's not that they're doing that to enjoy the scenery. It's that you are breathing so hard and you're just telling yourself, you know what, only take 2 breaths and then you got to take another step because you just feel like you can never catch your breath once you get high enough in there. So that's what limits how fast this can go. So again, you're not going to need to know what each of these intermediate steps are, but that you put acetyl-CoA in, adding 2 carbons. You oxidize those 2 carbons off, and for each glucose, you put 2 of these in. So at the end of this, you've completely oxidized and burned up all of those carbons. All of the carbons from your glucose have now turned into carbon dioxide for you to exhale. So making sense? Yep. So just like when you take your piece of wood and you stick it in a fire and it burns up and you get heat and energy from it, you take the glucose that you put in your mouth and you burn it up and get energy from it. And that fire makes carbon dioxide waste, so do you. So if you want to measure really accurately how much energy you burn, the way they do it, if they're looking to go, okay, how many calories are you burning while you do an activity, they capture the carbon dioxide off of your face, coming out your mouth and your nose, and look at how much carbon dioxide comes out. And that will give you really accurate. When you calculate out and say, okay, well, my body weight is this and all this kind of stuff to estimate your calories, you're estimating. There is always going to be some fluctuation person to person based on your own personal metabolic rate, your muscles. That capturing that carbon dioxide is the way to measure it most accurately. So now we have all of these NADHs that we need to do something with. And the way we're going to get their energy out of these is this electron transport chain. So these NADHs have been a way of harnessing these electrons. And again, all of the steps are written out on the side, but we're just going to go straight for the picture. So inside of your mitochondria, you've got this membrane on the inside, and these are proteins that are embedded on that membrane on the inside of it. So mitochondria, that kind of sausagey looking thing, your powerhouse of the cell, it's in these membranes in here. So the more mitochondria you have in a cell, the more energy you can make, because that's where this is coming from. So we have these different protein complexes here, 1, 2, 3, and 4. And they've got these different molecules inside of here, the FMN, like flavomononucleotide. This is riboflavin, one of your vitamins is used for this. A lot of the B vitamins are actually used to make the enzymes that are the intermediate steps of metabolism. So when people say, oh, I feel like I have more energy taking a B vitamin, what it is it's not that they've actually gotten energy from the vitamins. It's that the vitamins have allowed them to have all of their enzymes working. However, it isn't like you can say, well, if I got a little bit of energy from taking this much, I'm going to take a whole lot of them. Once you've got all your enzymes working, it does you no good to take any extra vitamins. Then they're just going to get flushed out. If you keep taking more and more and more, you can actually end up with toxicity from it. So determining what is exactly how much you need, again, that's one of those things we can approximate what a human needs. We can't 100% say today you need this much of this one, because you never know what your activity level is actually going to end up being at the end of the day. So NADH is going to start here, and it's going to pass its electrons here at number 1. And you'll notice they draw this kind of going downhill. So as it moves through here, these electrons get moved down the chain, and they end up coming out at the other end. FADH2 kind of cheats. It dumps its electrons off partway down the hill. So if you roll a ball from the top of the hill and you roll another ball from like partway down the hill, are they both going to have the same kinetic energy? No. This one's going to have more because it's released from here. So you're going to get more energy, more ATP out of NADH than you do FADH2, just because you don't drop the stuff down the hill as far. And again, all of these have names. This one, iron sulfide in here. This is one of the reasons why you need to have iron intake, is iron is going to be good for being oxidized, going between an oxidized and a reduced state. So when it's in your hemoglobin and it's got oxygen in there, it's going to be in one state. When it gives up the oxygen, it's in another state. Same thing in here. When it takes the electrons, it's in one state, and it will go back and forth. So if you have iron deficiency anemia in there, inadequate iron levels will make a person tired because you're not going to be able to use as much of this chain in there. And so there are certain things that could actually come in and replace iron. Certain metals could slip in here and replace iron, and they're poisonous because they don't do what they're supposed to do in here, and you'll basically destroy all your electron transport chains and get no energy and die. So now that you see that they move in this downhill method in terms of energy, now we're going to look at them folded in a membrane. So you got 1, 2, 3, and 4 here. As you move the electrons on here, what this causes these protein complexes to do is release a proton that we indicate with the H+ here onto the other side of the membrane. So what you end up with are these protons all separated from these electrons here. They don't want to be separate. They want to be together. Positive and negative want to come together. And it passes them along the way because when they get down here to the very end, you have this enzyme, this ATP synthase, that is going to allow them to come together. And when they come together, well, they're actually coming together here a little bit too. You've got the hydrogen and the water coming together to make oxygen. And when they come through here, it gives you the energy to glue a phosphate onto ADP to make more ATP. So the more electrons you have, the more protons over here, the more ATP you get to make. And when this comes through, they've actually looked at it on a microscopic level. How they looked at this to determine that it works this way, I don't know. I think it's pretty amazing that we can figure this out, though. So you've got your inside membrane here, the space where these hydrogens have come through. And the way this protein is actually shaped is you've got this rotor and stator. The hydrogen drops down through something in there into a little spot on the rotor. It actually spins around, and when it gets around to the other side, it gets dropped through the next slot and comes out. And this spinning in here generates the ATP. So this actually works like you have mechanical rotors and stators that they've built to harvest energy. And we built these things without knowing what this thing was in nature. It's kind of interesting what nature actually gave us in our cells when we look at it now is the same thing that we built. But they're looking at, okay, well, if nature has these in mitochondria, how could we use this to somehow use mitochondria to artificially create energy outside of humans for creating power in there using this mechanism? So for every NADH that's going to go through, they figure that it's not actually a whole number in there, and in here it gives you this 2.5 to 3.3 ATP made. we round it to 3 when we count these. The FADH2, we round to 2 when we count those. So when you look at all of those that go through, what you end up with is 38 ATP. So then you take away your 2 that you had to invest in glycolysis and you get 36. FADH2 is like it's like the NADH that's going to carry electrons. So this is another one of those pictures that kind of summarizes things. So from glycolysis, that first cycle where we split the glucose in half, you have a net gain of 2 ATP. You convert it to that acetyl-CoA, stick it into the citric acid cycle. You get 2 ATP out of this. And then you have all of these NADHs and FADH2s. All of these end up coming through the electron transport chain to give you your 26 to 28 ATP. So if you want to remember numbers in there, because 36 is the number that you'll see most of the time on tests where they talk about, okay, you get 2 net gain ATP out of glycolysis. Your key numbers that you'll want to remember. Glycolysis is going to give you a net gain of 2 ATP. And then the citric acid cycle and the electron transport chain together. I'm sorry, what was that? Yeah, it's your energy currency. So these two together will give you 34 ATP. These are your averages. Sometimes it is a little more, sometimes it is a little less, but your averages that you get tested on standardized tests, go with these numbers. So the citric acid cycle alone is 2 of them. All the rest require both of these 2 to get the ATP. So when you look at the complete oxidation of glucose under standard conditions, it has a negative 686 delta G. So when you phosphorylate ADP to make ATP, what you're going to get is 7.3 kilocalories per mole of ATP. And then when you figure, okay, you've got 38 moles of ATP that are produced over 686 kilocalories per mole, you end up with about 40% of the potential chemical energy in glucose being transferred to ATP. That is when you pull off one of the phosphates. So ATP is like a full wallet, ADP is like an empty wallet that you can fill back up. So under cellular conditions, they figure it's probably a little bit higher, but under lab conditions, it's about 40% efficient. And while we look at that and go, oh, only 40% efficient, we aren't real good at making anything mechanical that's even close to that efficiency. So 40% is actually pretty good in there. So what happens to the other 60% in there? A lot of it gets used to maintain your body temperature. And you have to keep warming back up your body because if it gets a little too warm, it sweats in there. So that's what ends up happening to it, is it keeps you warm. So even the most efficient car, and I think this is probably including things like a Prius, is about 25%. So let's go ahead and take a break for a couple minutes before we talk about fermentation. On with fermentation. So fermentation is what's going to occur when you don't have oxygen present. So we do a little bit of fermentation in some of our cells. We can do the glycolysis when oxygen is not present, and then we can pyruvate, in order to go into the citric acid cycle, has to have oxygen present. If it is not, then it will go into a fermentation cycle. And there are several different fermentation cycles. They're usually much shorter in terms of steps, but what makes something fermentation is that the electron transport chain doesn't use oxygen at the end as its final electron acceptor. So instead of putting all those hydrogens with oxygen to make water, it puts them with something else. So an example with certain anaerobic prokaryotic bacteria, they will combine it with sulfur instead of water. So it makes hydrogen sulfide instead of water in there. So if you are around anaerobic bacteria infections, they characteristically stink. This guy with the sulfur, he's going to stink. So that is why anaerobic bacteria tend to stink, as they do this fermentation and they have to use other things like sulfur in there. So most of them are not going to smell real pleasant in there. So types of fermentation. So alcohol fermentation is another type that can be done by certain bacteria in there. Here it's the pyruvate releases the carbon dioxide to make acetaldehyde. And then you take the pyruvate and the acetaldehyde gets reduced some more with NADH to make ethanol. And then it will regenerate its supply of NAD+ so that it can continue to do glycolysis. So it's kind of like you have glycolysis that goes on, where you're going to take glucose to make pyruvate. This is going to require having NAD+ that gets converted to NADH. You have to have more of this. So when you take the pyruvate and convert it to ethanol, you're also going to make more NAD+ to come up and keep feeding that cycle. So that you can keep doing that. So this is not completely oxidized. Ethanol, if you remember back from that chapter where we talked about the meth-eth-prop-bute, this still has 2 carbons in it. So you didn't really break down that much from this. But in the process, you did make a little bit of carbon dioxide in here. So if you have things fermenting in your gut, up higher in your bloodstream when you are doing the normal metabolism in your cells, you take this carbon dioxide and you exhale it. If you have bacteria in your gut that are fermenting things, you're going to put this in your gut and it's going to come out the other end. So lactic acid fermentation, this will occur in your muscles. So when you say you go out and you're going to do a run and you decide, you know what, I'm going to sprint. I'm just going to go as fast as I can. What's going to happen is really quickly, you're not going to be able to breathe hard enough to sustain that, and your body's going to want to slow down. But before it slows down, what's going to happen is some of the muscles are going to continue to try, and they're going to have all this pyruvate that they've built up from doing glycolysis. And what it will do is get converted into lactic acid. So You take the pyruvate, you make lactic acid. It resupplies the NAD plus to keep the cycle going in there. So lactate is lactic acid. Chemistry likes to give things multiple names too. So it used to be thought that the lactic acid was the main contributor of delayed onset muscle soreness, and they don't really think that's so much the case right now. It ends up you don't hold the lactic acid in there as long as you thought, and they've done studies where they actually neutralize that acid in there, and that the delayed onset muscle soreness is probably more related to changes in the ions and then being able to also have microtrauma to your muscles that will cause the soreness. So this lactic acid, though, that you have in your muscles, you can actually send that in your bloodstream back to the liver, and it will convert it into pyruvate that you'll have to break down later on. So when you have that lactic acid in your muscles, it is going to change their performance because it's going to change the pH, but you still want to try and flush that out of there. And mild aerobic activity is going to be the best way to do that, because that's going to tell your liver, okay, we need to have some more fuel, and creating the draw on the fuel is going to take this and have the liver do that for you. So does it make sense, the difference between fermentation and aerobic metabolism in there? Fermentation doesn't have nearly as many steps. You go basically acetaldehyde to ethanol or pyruvate to lactate. There are other things that are fermented, but we could spend a whole quarter looking at fermentation cycles and not cover them all. So in your muscles, and people usually care about this one because ethanol is what you drink. So this compares them side by side in a cell. Both start out doing glycolysis to make the pyruvate. So if oxygen's present, pyruvate goes into being acetyl-CoA in the citric acid cycle. If oxygen's not present, it's going to do the fermentation pathway. So aerobic respiration is far more efficient. It's going to be 19 times more efficient. It's going to give you 19 times more ATP than fermentation does. So we have some different categories of organisms. Obligate anaerobes. These can only do fermentation. So these are going to be bacteria that are in this category. And they cannot survive in oxygen. So they don't have the enzymes to help process the oxygen. Oxygen will oxidize them to death, literally. Obligate aerobes, these are going to carry out aerobic respiration, but not fermentation. So that's the only way they run, is doing aerobic respiration. The human brain, it's a vertebrate, has to do aerobic respiration. It will not do fermentation to keep your brain going in there. So if you don't have oxygen present, your brain is not going to work. Facultative anaerobes, these can make enough ATP to survive using either fermentation or respiration. They will prefer to use aerobic respiration because it's more efficient, but if they have to, they will do anaerobic metabolism. And your muscles are this way. They would prefer to do aerobic metabolism, but they'll do anaerobic if they have to. So evolutionary significance of glycolysis. It's thought that the ancient prokaryotes probably used glycolysis to make ATP before oxygen was present in the atmosphere. Once we started having the cyanobacteria that were able to produce oxygen gas as a byproduct of photosynthesis, then we had oxygen in the atmosphere and we would have developed the aerobic metabolism. But there was no aerobic metabolism before there was oxygen in the atmosphere. So glycolysis, we look at it with glucose, but you actually can have a wide range of carbohydrates that will come in. So here we have glycolysis. So any other carbohydrate is going to get broken down into its simple sugars, and it's going to join into somewhere in this cycle early on. Most of the time it's going to come in somewhere between glucose and glyceraldehyde 3-phosphate. So if you eat fructose, it's going to come in here. If you eat lactose, you're going to break that down into your galactose and glucose. So all the different things that you will eat will get broken down and enter into this cycle. With proteins, they're going to first get digested into becoming their amino acids. And what the body likes to do with those is take those amino acids to build new proteins. It does not want to use this as an energy source. So a lot of times people go, oh, I need to get some protein for some energy. No, this is what your body wants to use for energy. The amino acids, it will do that when it has to do that for energy. But it's also going to produce byproducts called ketones that are going to start to make you smell. They will, yes, they'll suppress hunger, but most of us don't eat because we're hungry. We eat because it's there. Yeah, I mean, if you just look at how many times you walk by your refrigerator and open the door, when you know you haven't gone to the store, nothing's changed. We eat because it's there. And so suppressing your appetite doesn't help you a whole lot. So it will burn the proteins when it has to, but it does not want to. It would prefer to use carbohydrates for that. What it is going to do, though, is it's going to create this amine waste that you have to eliminate through your kidneys. So if you have kidney problems, you probably don't want to eat excess proteins because anything that your body is not using to rebuild structures has to be processed through the kidneys. This ends up becoming uric acid, which can make crystals that are kidney stones. If you eat excess amino acids, some of that goes in there, but some of it is also going to go to acetyl-CoA here. And what it doesn't show you is that this pathway is actually reversible. So make sure I didn't skip too many. So a lot of times people get told with the fad diets that, oh, well, you just eat as much protein as you want and you won't get fat from it. Yeah, you will. So what's going to happen to fats that you eat? Most fats you eat are going to come in the form of a triglyceride, which is going to get split into glycerol that comes in and can be broken down into this pathway, and your fatty acids that come in at acetyl-CoA. So you've probably heard that you have to have oxygen present to metabolize fats. Because they come in at acetyl-CoA and to this part that is your aerobic metabolism, this is why they say you have to have oxygen present to do that. So when you exercise, what you're doing is you are causing this part to spin faster so that you can put more into this. So it isn't that you only burn them when you are exercising. Right now, this is your body's favorite form of fuel. It's going to take from this as much as it can. You guys can come in and look at slides. But if you don't have, they're on top of the thing over there. And just look around for microscopes. I didn't put them away, so I don't know where they are. So when you get down to here with the pyruvate in there, if there is extra, it will come here, it gets converted here. But it will use the fats right now if there isn't any of this to use. If there is too much of the protein in there, it isn't even going to begin to touch any of this. So that's the other problem with saying, oh, you can eat as much protein as you want. You're going to use up all of this before you even start to attempt this pile. The body looks at, well, which pile does it cost me the least to store? This one doesn't really cost anything to keep it on there. It's lightweight. It doesn't do anything. It doesn't need metabolism to keep it alive. Muscles do. So it will store this very happily. It loves to store this. So when you break off the fatty acids, most of these are 16 to 24 carbons long. Acetyl-CoA is only 2 carbons. So you're pulling off 2 at a time. One of the reasons why fats have so many more calories in them is if you've got 3 fatty acids, each of them with 16 carbons, and you pull off 2 at a time, that's going to be 8 from each of those fatty acids, 3 fatty acids in 1 triglyceride. That's 24 turns of the cycle to break down one of your triglycerides, where it's only 2 to break down 1 glucose. So this is a really efficient form of storing energy, is to store it as fat. Stores lots of energy with relatively little space. So Anabolism Biosynthesis. So you can use some of the carbon skeletons. Some of the things around in here can actually be pulled off and be used to make other things in the cells if you need them. So that would take away from some of the energy you would get from something. If you are in a place where starvation is a concern, you know, then you might be running short on energy and you're going to tap into your fat stores. However, in this country, we're really not looking at any shortage of food for most of us. So we're going to have plenty of stuff to pull off of there. We're not going to need to take away from our energy stores. So regulation of respiration through feedback mechanisms. So the most common is feedback inhibition, where the products of the anabolic pathways inhibit earlier enzymes. If you're making substances you need in your body, when you have enough of it, that end product that you make is going to shut down the process in there. So that is the end of glycolysis. I'm going to start a separate